Hello, my name is Stella Planou, and I'm a senior scientist here at Residual. With me today is Cathy Frankis, who is a technical expert in chromatography and spectroscopy. Today we'll go through the use of ion chromatography in pharmaceutical quality control. Before we start, let me give you an overview of RSSL, who we are and what we do at Reading Scientific Services Limited. We are a cutting-edge contract-based research organization pushing the boundaries of science and innovation to support our clients developing life-changing treatments for patients. Our clients trust us to deliver innovative solutions to real-world problems facing the global life sciences, pharmaceutical, healthcare, and personal care sectors. Here in Reading, in UK, our multidisciplinary team works together with our clients to develop drug products that they are safe, innovative, and capable of transforming lives around the world. We offer a diverse range of biological, microbiological, chemical, physical, analytical services, bespoke training and consultancy to deliver tailored solutions. We are exceptionally proud of the depth and breadth of expertise which has been strengthened and shaped by 30 years' experience of working in partnership with pharmaceutical and life sciences sector. Over this period, we have built an extensive portfolio covering a wide breadth of analytical techniques, of which we have detailed some here, like chromatography techniques, mass spectroscopy, elemental analysis, spectroscopy, microscopy, etc. Let's go through the index of our presentation. Firstly, we will speak about the use of IC pharmacopeia and examples of IC applications from monographs, and then we will continue how monographs can be the start of method development and the importance of verification and validation. The regulation of pharmaceutical ingredients and raw materials is crucial to ensuring treatment efficacy and patient safety around the globe. One of the many ways this is achieved is through the use of regional pharmacopeial testing in compliance with published methods to allow materials to be assessed against set specifications. Also, general chapters should be consulted. In addition to the above, in pharmacopoeia-specific monographs for identification test, assay, and impurities are included. In more details, an identification test, as some of you might already know, is used for the identification of compounds using reference standards by comparing their retention time. Assay is used to determine one of the main components in a sample, and impurities to determine the purity of the sample. Furthermore, an assay can provide a quantitative measurement, while impurities can be indicated quantitatively or qualitatively. Something else that should be mentioned is that IC affords increased selectivity and sensitivity over traditional wet chemistry techniques, ambiguity, and removal of subjective readings. Someone could ask why you are not using HPLC instead. HPLC is one of the most powerful analytical techniques in the modern laboratory. From research to manufacturing to legal, it is widely used in many sectors. However, there are some limitations. Ionic and polar compounds pose a challenge, so do organic acids. Moreover, some analytes, such as fluoride and chloride, are not active in the UV range, making it impossible to determine them with a UV detector, the detection which is most commonly used with HPLC. Ion chromatography, on the other hand, is ideal for the separation and detection of ions and polar molecules, both organic and inorganic. It has the capabilities to analyze from small molecules to larger biomolecules like organic acids, carbohydrates, amino acids, peptides, oligosaccharides, and proteins. This makes IC the ideal extension of HPLC, increasing the analytical capabilities of the modern laboratory a lot. IC has been applied to all aspects of the manufacturing and disposition of pharmaceutical products, including characterization of active ingredients, excipients, impurities, raw materials, and formulated products. The majority of IC methods use either anion or cation exchange chromatography coupled with suppressed conductivity detection. IC is especially valuable for ionic or ionizable analytes that they have little or no native absorbance on UV. In addition to suppressed conductivity detection, the ion exchange separation can be coupled to other detection strategies, including pulse barometric detection, UV absorbance, and mass spectrometric detection, providing a wide range of analyte sensitivity and specificity. The wide range of the majority of the IC detection methods makes IC applicable to the quantification of trace contaminants as well as major product components in the same run. IC typically uses dilute acids, alkalis, buffers, or salt solution as mobile phase, reducing solvent cost and simplifying disposal logistics. In most cases, the eluent can be disposed of after appropriate neutralization and, when necessary, after dilution with water. Most of the IC methods are using automatic eluent generation, which brings many benefits to the lab. The eluent generation improves the analytical consistency and reproducibility day to day, system to system, lab to lab. It provides uniform, highly stable eluent concentrations. Also, it gives highly reproducible results with little variability in pin detection times or areas. Apart from the above, enables gradient separation with an isocratic pump and is also a very safe technique, as no manual preparation is needed, so no handling of strong acids and bases. Now I'm handing over to Cathy to take you through some examples and applications.
Thank you, Stella. My name is Cathy Frankis, and I'm a technical specialist in chromatography and spectroscopy techniques here at RSSL. Today I'm going to take you through some examples of ion chromatography applications from pharmacopeial monographs and illustrate how and why they can form the basis for development of new analytical methods. The first example shows an identification and assay test for fluoride ion in stannous fluoride as per the USP monograph for this material. Up until recently, the identification of this material was conducted using a wet chemistry method where the test solutions were added to calcium chloride and a white precipitate of calcium fluoride was observed. The move to IC provides an improvement in selectivity and removes the subjective nature of the chemical test. The test method for IC uses conductivity detection in suppressed mode and stipulates that the use of an automatic eluent generator is permitted, which provides more robust chromatography with less chance of peak drift throughout the sequence and avoids the need to prepare eluent manually. Interestingly, this monograph stipulates two alternative column codes are acceptable for this monograph. Columns of the type L113 stipulate a specific chemical description relating to particle size of 7.5 microns with a precise level of cross-linking. The alternative column type, L91, allows for a more general definition of monodisperse beads coupled with quaternary amine, with a much broader range of bead size between 3 and 10 microns. This allows for a wider range of column technologies to be applied, including more modern high-pressure columns, often with smaller particle sizes that allow for improved resolution. In this case, the USP has future-proofed the methodology against future advances in technology. This slide shows another example from the USP, a limit test for a known impurity galactosamine within sodium heparin. The sample is hydrolyzed in hydrochloric acid at 100 degrees Celsius for 6 hours, resulting in the breakdown of the polysaccharide. The test checks that the concentration of galactosamine observed under these conditions is less than the permitted specification. Pulsed amperometric detection, or PAD, is used for this test. The monograph stipulates the waveform that should be used, but not other details, for example, the type of electrode, which means there is some flexibility in hardware configuration. Again, the column class is specified in the monograph as L69, but in this case, the USP specifies that a PA20 will meet the requirements, avoiding any ambiguity or comparison between column parameters to be needed. An amino trap column is specified as well as a guard column to remove any amino acids which can interfere with the carbohydrate analysis. In this case, the monograph does not explicitly indicate that the use of an eluent generator is permitted. Therefore, it will be necessary to seek client agreement and, if necessary, verify the method to show the equivalent results can be achieved with and without automatic eluent generation. Here we have a more established example of conductivity detection being used, this time in the limit test of ammonium in sodium bicarbonate. Eluent generator cartridges are available for cation analysis using MSA cartridges, as well as anion analysis. And fortunately, this monograph does stipulate that this mode of eluent preparation is acceptable. The monograph is quite unusual, as it uses an elution gradient to include a wash step. Most monograph methods are isocratic. However, a runtime is not provided in the method details, and therefore some trial and error was required during the method setup to ensure the system was re-equilibrated prior to the next injection. As shown in the figure, the biggest challenge here is that the ammonium peak elutes on the end of the tail of the large sodium peak, and particular care needs to be taken of the column and suppressor to ensure the system suitability criteria for the method can be met. The column needs to be well washed before and after each analysis, and special attention paid to the suppressor to ensure it is well hydrated. The monograph also recommends a 2mm suppressor be used for this application. In instances of suboptimal chromatography, we have found the suppressor is the first thing to check. A rupture in the membrane will have a detrimental effect and may mean your system suitability criteria will not pass. As the test is a limit test for ammonium, which is fairly prevalent in the environment, we have found it is advisable to limit the use of glassware as much as possible and use separate vials for each injection, especially during long sequences, to limit the dissolution of ammonia from the atmosphere. In 2015, the European Pharmacopoeia moved from the traditional thin layer chromatography method of assessing ninhydrin positive impurities within amino acid raw materials to stipulating that a chromatography-based technology must be used. RSSL worked with Thermoscientific to find a solution which achieves chromatographic separation with ion exchange columns coupled to a post-column derivatization instrument that introduces ninhydrin to the sample after separation is achieved. Quantification is then achieved by visible absorbance detection, and the method provides more certainty in the analytical results compared to TLC, which can be subjective and can lack the required sensitivity. As the method is detailed within the general chapter for amino acid analysis, and specific chemistries of eluent or column details are not provided, it was necessary for us to verify this solution for the entire range of amino acids which required the testing before offering this testing to our clients. This obviously incurred a significant time investment on installation, but allows us to be fully compliant for all our customers without the need for separate client or material-specific verification exercises. One of the system suitability criteria which some other instrument setups can struggle with is the resolution between leucine and isoleucine, but as you can see, these analytes achieve the 1.5 criteria value set by the EP easily. Setting up methods from the pharmacopoeia can be challenging because provided method details are often very sparse. In the first instance, it is important to make sure you have all the necessary hardware, consumables, and reagents. Do you have the right mode of detection? Do you have a pump capable of delivering an elution gradient, if it is needed? Do you have or can you buy the required column and/or suppressor? In this instance, it is worth remembering that the general chapters within different pharmacopoeia will provide guidance on some column parameters where variations may be acceptable. For example, flow rate can be adjusted following a change in either particle size or internal diameter of a column. It's important to plan and conduct a feasibility study to ensure chromatography that meets the requirements of the test can be generated. Attention should be given to any provided system suitability requirements, as well as the observed resolution between peaks, the lack of interference by running system and diluent blanks, and the injection repeatability should also be assessed. Sometimes the pharmacopoeia methods provided really are not suitable, possibly because the monograph is older, or perhaps there is a detail that is crucial that has not been included. In this instance, check for application notes, which may try to address the problem. Perhaps there is an application note, but it uses a completely different mode of separation. In this case, double check whether this alternative method might be suitable for your application, but bear in mind that a move to any method details that do not agree with the monograph method will mean that either method verification or validation will be required. In general terms, verification is a check that a previously validated method for an analysis in a matrix is performing correctly using a particular instrument setup, whereas validation is the more encompassing process of assessing an alternative methodology as being suitable for a particular application. Just to illustrate this point, this slide shows a summary table including the parameters that should be assessed for an identification, assay, impurity limit, and impurity quantitative test for method verification and validation, respectively. These are in line with the requirements stipulated by the USP, and for validation also line up with the expectations set by the International Committee for Harmonization of Compendial Methodologies. You will notice that the number of parameters increase with the complexity of the test and reflect the requirements. For example, an assessment of LOD is required for limit test, whereas LOQ, or limit of quantification, is required for quantitative assessment. For verification especially, there is the flexibility to add or remove parameters to meet a client's cost or timeline requirements. All testing plans for either verification or validation should be contained within a protocol document signed by both the client and the analysis provider prior to commencement of the exercise. Monographs supplied by the pharmacopeials are designed and validated for specific raw materials. So what happens if you need a method to assay or test for a related substance in a drug product which will contain a mixture of ingredients? Monograph methods can be a great starting point for method development. For example, the fluoride assay method presented earlier may form the basis of a fluoride assay in a toothpaste or tooth gel matrix. However, there are a number of factors that should be considered when applying monograph methods to finished products. Will the method provide sufficient sensitivity for the analyte level in the product? How much target analyte is present? Will any other ingredients in the product generate peaks which could interfere with the chromatography? If you see an interference using an isocratic gradient, think about switching to an elution gradient to see if you can improve the separation between your peaks. Can you use solubility differences within your matrix to improve the sensitivity of your method or remove the components that are resulting in the interferences? Finally, consider whether there is a need for a wash step to remove matrix components which may become stuck on your column, and when including a wash step, remember to include an equilibration step. Remember that even if your method is based on a monograph application, as it is new for that particular matrix, full validation of the method will be required to ensure reliable, robust results be generated. Within the pharmaceutical industry, there is a need to assess materials for a range of residual solvents, depending on the specific synthetic pathway that was used in the manufacture. As such, monographs cannot specify which residual solvents should be considered for each material, and instead a general chapter provides guidelines as to applicable residual solvent limits based on the associated toxicity of each. Gas chromatography-based methods are the industry standard for residual solvents analysis due to their volatile nature. However, certain acids, for example acetic and formic, can prove to be a challenge for these techniques due to sensitivity issues. Ion chromatography with conductivity detection can be used as an alternative approach in these cases. However, as such, the USP stipulates that these methods must be fully validated for each matrix to which they are applied. The slide shows some example chromatography from a traditional ICE AS1 column, which was selected due to the surfactant nature of the matrix. The method is quite old-fashioned and uses chemical suppression rather than electrolytic and requires a separate pump to deliver the regeneration solution. However, the resulting resolution between the acetic and formic acids is very good, and generally this approach produces less interference issues for some types of materials. In recent years, there has been an increased focus on analysis of low-level nitrite and nitrate in pharmaceutical APIs and excipients due to their mechanistic role in the formation of nitrosamines. Ion chromatography has long been used for anion screening, and new advances in column technology and high pressure systems mean that we are now able to see levels as low as 1 part per billion on column. In general, this method allows for a straightforward preparation step where the sample is either dissolved in or extracted with milky water. As this approach is a new methodology, validation will be required where the generated results are to be used within any kind of regulatory submission. However, verification may be sufficient if the work is required only for investigative purposes, for example, into the driver for nitrosamine generation within a particular product or type of products. One disadvantage is shown in the figure, where a material containing a high level of chloride, which elutes just before nitrite, is interfering with the nitrite peak, which will affect the method of accuracy and sensitivity. However, this can be solved by moving to an alternative mode of detection, either UV or mass spectrometry. In conclusion, I hope within this presentation that we have been able to show the critical role the pharmacopoeia play in ensuring strict regulation of raw materials and finished products within the pharmaceutical industry. We have discussed the importance of monograph methods in ensuring this regulation, but that they can also be a useful resource of knowledge to promote good working practices for the technique. We have looked at the prevalence of ion chromatography within the pharmacopoeia monographs, with over 70 monographs featuring an IC method in the USP alone. These methods feature a range of detection modes, such as conductivity, pulse amperometric detection, and UV visible absorption. It is likely that the number of methods based on ion chromatography techniques will increase in the coming years as the industry moves away from subjective wet chemistry techniques and more readily embraces more technically advanced approaches. Monograph methods are actually a great starting point for method development, but a number of factors need to be considered, including the sensitivity that is required, as well as the impact of other components in the matrix which may cause interferences in the chromatography. Finally, once you have a method, it is important to show that it is suitable and meets the necessary requirements. It is important to verify where a previously validated method is applied to a new matrix or uses alternative hardware. When an alternative method has been employed to that stipulated in a monograph, full validation of the method must be conducted. And with that, all that remains for me is to thank you for your kind attention today. I hope you enjoyed finding out about ion chromatography in the industry.